So, but what we're starting to talk about here is a little bit, obviously we started out pretty narrowly talking about reproducibility in the lab and specific examples of that, which is bread and butter for scientists, right? But the context is really also very important. You mentioned the need to keep your career alive and going, and the funding issues. I mean, these are huge overarching issues that, so I mean, we're looking at this on a bunch of different scales, and I think it's important to remember that whole context as we're having this conversation. But I do want to go back to the sort of down to the details just for a minute here and get back to sort of, if you guys could each offer sort of practical advice about, you know, you've described some of these problems, and are there some generalities you can offer to people who are going to have complementary problems, and how they can think about attacking them and solving them?
So for me, it would be the control of the microbiota through proper litter-mate controls, as I mentioned, and that's the het-by-het breedings. Again, it's costly. It takes a lot of time. Genotyping, all those things, it takes a lot of time and money to do the proper experiments. And as I mentioned, hopefully, if you're able to use the het mice as your control mice, then you're saving time and money on that. So that would be my advice, is to see if your phenotype is trackable in between the hets and the knockout animals.
And what proportion of the reproducibility or variability do you think you're addressing by doing that in your experiments?
For us, I'd say it's the microbiome has such an enormous impact, as I mentioned, in gut immunology, like we study.
That makes sense, yeah.
But you know, we've seen it in a lot of other different contexts. Like I said, behavior and other, I'm sure in sepsis models as well. There's a huge impact on how the microbiome has impacted on the immune system of those animals and how they then respond to a challenge.